In this video, we will discuss prokaryotic transcriptional regulation. The four modes are positive inducible, positive repressible, negative inducible, and negative repressible. Today, we will be analyzing the negative inducible mode. Let's first go over what the term negative inducible means. If a mode is deemed to have a negative control, then the regulatory protein acts as a repressor and binds to DNA to prevent transcription of the structural genes. Remember that structural genes are genes that encode for proteins used in metabolism or that play a structural role in the cell. The latter part, inducible, means that transcription is normally off but is turned on when a small molecule binds to the regulatory protein. This will be easier to understand if we look at it in action. Here's an illustration of the negative inducible mode in the absence of a small molecule. Some key features to notice on the operon is the promoter region, which is the landing pad for the RNA polymerase, and the operator region, which is where the repressor protein binds to the DNA. Also, note that the repressor protein comes from the repressor gene that encodes for it. So in this case, when the inducer, another term for a small molecule, is absent, the repressor protein is able to bind to the operator. This inhibits transcription from occurring by blocking the RNA polymerase from moving down the DNA, meaning transcription is off. When a small molecule is present, it binds to the repressor protein, which inhibits it from binding to the operator region of the DNA. Since the repressor protein cannot bind to the DNA, the RNA polymerase is not blocked, in which case it can freely move down the DNA to carry out transcription of the structural genes. Even after illustrations, it can be hard to get it, so here's an analogy that represents the negative inducible mode of transcriptional regulation. Let's say your ball accidentally went into your neighbor's yard, where their big, mean dog is. In this case, getting the ball represents transcription. The dog represents the regulatory protein. A dog bone represents a small molecule, and you represent the RNA polymerase. When there is not a dog bone present, the dog blocks you from getting to the ball. In essence, transcription does not occur because the regulatory protein represses the RNA polymerase from moving forward. However, when a dog bone is present, the dog has a higher affinity for the bone than blocking your path to your ball, meaning you can freely walk and retrieve your ball. In other terms, the small molecule induces transcription by binding with the regulatory protein, thus enabling the RNA polymerase to move down the DNA and allowing transcription to occur. These are my references.